Fox with Inscripta. And I'd like to start with a little bit of, I guess, what they say in the comic world, an origin story. So I was originally trained as a nuclear engineer, and before you run away in horror, like, what can this guy possibly have to contribute? That's only the beginning of the story. It turns out, as fate would have it, I, sometime in the mid-90s, found myself, of all places, aboard an aircraft carrier somewhere out in the Pacific, where I had stumbled across a book that was lying around called The Selfish Gene by Richard Dawkins. And I, along with I think a lot of other folks who've read that book, that had a profound impact on my life. It changed the way I thought about the world and really instilled in me an incredible sense of awe and appreciation for the power of evolution. So that experience changed the course of my career, and I eventually found my way to playing around with proteins using directed evolution and working on new tools and strategies to accelerate that process. So the experience was incredibly gratifying, and many of the friends and colleagues I developed over the years at companies Maxigen and then Codexis, who developed these strategies and tools, eventually were found left somewhat wanting in the sense that we had a desire to work with larger biological systems, namely pathways and genomes. So that was the aspiration at the time. Unfortunately, the tools were inadequate to carry that out in the ways that we had hoped for. So I had to wait a few years, but I had the very good fortune a couple years ago to meet some folks who had a vision and a set of technologies that would, or had the promise to change that. So those folks, along with a number of others who've joined the organization since then, are all now part of the company Inscripta, which I'm very grateful to be a part of. So today, really for the first time ever publicly, we're going to share with you some of the results of the very hard work and amazing progress that team has been able to accomplish over the last few years to really further the goals of biological engineering. And I think more importantly, based on that progress, we are readying solutions to share with you in the synthetic biology community that we believe will enable and democratize genome-scale engineering. Okay? So in terms of what we'll talk about here is first kind of a setup, and it's kind of forged in the fires of the directed evolution protein engineering work I did for many years. And then we'll talk about how to extend that to pathways and genomes, and then highlight some of the issues and challenges associated with that. And then, of course, we'll share with you what the Inscripta vision is, or the solutions for addressing those challenges. And then I hope you'll find some of the interesting demonstration application and the surrounding data to be instrumental in sort of firing your own imagination for how you want to use this technology. And then really kind of wrap up with what's next, not just for Inscripta, but more importantly for you, because we are really trying to set up a platform for you, the community, to be able to accelerate your own genome engineering research. Okay, so genome engineering. This is our aspiration, but it's difficult. And we heard a wonderful keynote this morning from Ginkgo talking about how difficult it is, and it's essentially because we don't know what the rules are, like building a computer or an aircraft carrier, as it were. The rules are very hard to pin down. We don't know what many of them are. There's a lot of instability in the system, a lot that we don't know. And so predicting a priori from first principles how to design a system from the ground up is sort of in the distant future. So that's the going in ethos, that you, for the foreseeable future, will be locked into an empirical approach to letting nature tell you what works and what doesn't, but hopefully guided and accelerated in a way to move faster through that process. So as I kind of alluded to in the opening remarks, the strategies and tools for doing this in proteins are actually very well developed. And two key aspects that have sort of been identified over the course of time are the notion of diversity generation and combinatorial optimization. So diversity generation is this notion of sequence space is big, how do you even know where to intervene in order to move the bar on the property or phenotype of interest? And then once you identify individual components in your system that are important, how do you then optimize in a combinatorial fashion over large combinations of that system, okay? So the question is, what's missing, right? How do we precisely engineer pathways and genomes to really unleash the power of biology? And not surprisingly, the piece that was missing, and we think we have some solutions here today to share with you, is the ability to do genome-scale writing, okay? So before we get into that solution, though, let's just pause a little bit and talk about, how complex that search space looks like. So whether you're looking at a protein or a pathway or a genome, if you've ever run the numbers, and this is almost obligatory for almost any protein pathway or genome engineering, introductory paragraph or presentation or slides, is that when you multiply out the combinatorial complexity, the numbers become stupefyingly large. It's impossible to imagine solutions that would search that space exhaustively. So we need to have some other strategy or algorithm to get through this very challenging problem. So, as the second E in seed characterizes, it's about evolution, right? We can take great inspiration from evolution that's been going on for billions of years on this planet and thousands to hundreds of years in things like plant and animal domestication. And then, as we all are well aware, is the Nobel Prize going to Francis Arnold for directed molecular evolution last year. My daughter thought this was hilarious that Darwin evolved into Francis Arnold. It's great. So that's awesome. We can use the power of evolution to engineer these incredibly complex biological systems. But it's worth talking about two primary modes of evolution because I think still to this day it's not widely appreciated how different the two approaches are. One is an asexual mutagenesis process where you start with some ancestral strain or DNA sequence or whatever it is you're optimizing over, and you mutate it either random or a directed way, and you take the very best variant at that first round of evolution, and you decide to use it as a template for following rounds of an evolutionary approach to higher phenotypes. And you can do this round after round, again, just taking the best thing and mutating it again. It works, nature does it, again, for billions of years, but there's a much more powerful, rapid way to move through this space. And it's basically sex, okay? If you ever wonder about why sex is so great and why nature invests the time and effort into finding mates and so forth, it's because there is so much beneficial material, genetic material, that gets generated in the course of evolution. Sex is an ideal way to recruit and accrete that beneficial genetic material. And so the idea that we all know from our early days studying evolution is to mate the most fit, this is survival of the fittest, mate the most fit parents with each other, and make much larger jumps in your phenotype that you're striving for. And then you just repeat this process over as many rounds as you need to go to achieve your endpoints. So this is the high-level cartoon version of it. But a study that was performed by Pim Stemmer and colleagues, actually coming up on, 17 plus years ago, was inspired by this capability that Pim Stemmer had a notion to develop, which was called DNA shuffling. And he originally did it at the gene level to unlock this recombination process. But this study was carried out at the genome level, and I think is even more appropriate for this audience and for what we want to talk about here today. And it pretty much consists of approaching it very much like this process of generating diversity in the first sort of step, and then recruiting the beneficial diversity into additional rounds of combinatorial optimization. So these are those two principles that I mentioned on one of the early slides, right? And so that was good in theory, but one of our industrial partners at the time came to Stemmer and colleagues and wanted to know whether this really worked in practice for an industrial strain. So they engaged in a collaboration where they took an ancestral strain that had been taken from 20 years earlier and subjected to 2 decades worth of classical strain improvement, so classical mutagenesis using different techniques, and 1 million assays. And they compared it with a strategy that had been done recently at the time, using a diversification strategy, which again, at this time was just based on random mutagenesis, but then jumped into a hypersexual evolutionary process. And in the course of just 1 year and 24,000 assays, they were able to get to the same endpoint that took 20 years and 10 million assays. So an incredibly powerful demonstration of the ability for recombination to accelerate your search algorithm. So let's just spend a couple more minutes on the power of this approach and some of the important principles surrounding it. So after you generate this diversity, and if I were to come to you and say, I want you to search over those 50 or 100 or 200 variables or however many you have, any library of sufficient combinatorial complexity will almost always have a shape that looks like this in terms of the amount of investment that you make in screening that population versus the best thing you pull out of that, okay? That population. And so what you see is after screening the first few hundred or first few thousand variants from a library of incredible size, you get something that looks almost quasi-asymptotic. It looks like there's nothing better in the population. But in truth, there's things that are much better in the population. You just don't have the screening capacity or the selective capacity to be able to pull them out. They're these increasingly rare needles in the haystack, right? So it's sort of the odds of winning the lottery or getting struck by lightning that you'll find something way out here, okay, if you don't have the capacity to search through all of those possibilities. So the other strategy is the one I've already kind of empirically demonstrated, is to take a recombination-based approach where you just take the first 500 or 1,000 and then you just identify what the best variants are and recombine them, all right? So it's essentially taking this improbability problem and breaking it up into more manageable chunks. That's really the power of the sexual approach. And important it's to understand that you don't need high coverage or you don't need to screen deeply into this population to take advantage of this evolutionary process. Okay. So there's even one more version of this that accelerates the process even faster. And it turns out that if you're sitting here generating genotype and phenotype data, these are the days of artificial intelligence and machine learning and deep neural nets. With enough data, you can understand what the local genotype-phenotype mapping looks like and empirically interrogate it based on the empirical mapping you've arrived at to decide where to move forward in sequence space next. So let me just give you a brief sort of cartoon version of how that works. So again, sort of staying with the idea of improbability, as it were, Richard Dawkins gave it a book-like treatment in this book titled Climbing Mount Improbable. And the idea is taken from a metaphor that's been around for almost 90 years now, which I think probably many of you have seen in the course of your careers, is to think about genotype-phenotype space as a walk up a fitness landscape, where the coordinates on the east, west, north, south directions correspond to genotype, and the elevation on Mount Improbable is the phenotype. So the idea is actually really simple, is to take a set of sequences around some local portion of the sequence function landscape, and then develop a predictive model that takes as input sequence and outputs phenotype Y, okay? Once you have in your possession that map, that empirically derived map, you can then use it to drive towards higher gradient directions in the fitness landscape, okay? Very simple idea, very well developed in proteins. The aspiration is to eventually, of course, take this onto pathways and genomes. Okay, so let me show you an enzyme scale version of this, and I want to stress here, this was work that was done with some colleagues that I was associated with at Codexis a number of years ago. We're not yet to Inscripta solutions, but I think it demonstrates sort of the main properties of or aspects of the approach. And that is, we were looking to engineer a carbonic anhydrase to be very thermotolerant. And the way we did this is to first diversify the protein with standard methods in the field, to take and do saturation mutagenesis, where we look at every single one amino acid variant of this protein, which was around 200 amino acids in length or so. And those libraries are actually not that large. They're only 4,000, 5,000, 6,000 or so-ish for a typical protein. And what you find is that screening the first 30, 40, 50% from that population, you end up with not just a good winner, we had something 70-fold improved, but more importantly, you identify many beneficial mutations that serve as essentially evolutionary fuel for that combinatorial search process, right? So once you're done with that diversity generation round, you can then drop into that combinatorial search problem, right? And here, the libraries get much bigger, right? Because the combinatorial complexity is large once you start multiplying out all the options. So now you have these very large libraries, and you screen actually in absolute terms even less, and on a fractional term, far less, in this case less than 2%. and yet you're able to lock in huge gains in fitness, just like I showed in the genome example, right? And then you use that information in the genotype and phenotype data sets to help guide the follow-on searches. And then from there, it's sort of lather, rinse, and repeat, which is a process I'm not too familiar with these days, but I'm sure you guys get the point. And it works extremely well. We were able to get millions of fold improvement, continuing on in this process, occasionally rediversifying to recruit more evolutionary fuel. All right? So, again, can't overstate this, that high coverage is not necessary to achieve successful outcomes. Nature's been doing it for billions of years quite successfully. Okay, so that's protein engineering, and that's great. It works well, and most of that, all that work that's been done is almost entirely plasmid-based, okay? So now we want to move into the genome, not just with proteins, but with pathways and genomes, all right? So in protein engineering, you're generally making, a small size of edit, although you might make many edits, more than a single base pair change, but often the codon is the target, the unit of selection, if you were. But in terms of pathways and genomes, we want to go much bigger. We want to be able to do protein engineering in the genome, but we also maybe want to target other elements in the genome. We might want to target ribosome binding sites or promoters, or maybe even stretches that include both, right? So we need larger edits. And we need to do this at the scale of the genome, right? We want to be able to go after the entire genome to generate the diversity to take into combinatorial optimization, all right? So if we look at where the world is kind of today, and we characterize it in two sort of key axes, one is the number of genomic locations that you're going after in the genome, on the x-axis, and on the y-axis, you're looking at the variety of edit types. So beyond just, say, a single base pair change, these larger swaps, insertions, and deletions for like promoters and terminators. Then if you sort of bucket where we're at in terms of the applications and the technologies, you have protein and pathway engineering, kind of occupying a space where you can hit a few targets and you can go to high mutation rates, but you're kind of limited in terms of being able to do that across the genome at many, many loci. On the flip side, I think we've all seen some amazing developments just recently in the space of things like genome-wide base editing, right? Which really do allow you to go after the whole genome, but it's limited in terms of the edit type variety that you can affect in the genome, right? So base editing is a good example. You can do genome-wide knockouts and, of course, classical mutagenesis. But all of these forms of editing are limited in terms of the variety that they can confer. So where we really want to get to enable genome engineering and genome discovery is to unlock the full potential of edit type variety, these non-trivial edit types, as well as going genome-wide in terms of both the number of locations and the number of edits, all right? So effective solutions in order to carry this out are going to require unprecedented scale, superior performance, and greater access. Okay, and we'll talk about what those are like as we go through this a little bit more. But this is where Inscripta comes in, right? And I want to stress again, these are solutions that we're developing not just for us, and in fact, not really for us, but for you in the synthetic biology community. Okay, and we'll talk towards the end about what that's going to look like for you guys, all right? So, how does this work? So just very briefly, if you look at today with the CRISPR technology, I won't go through the basics because I think most of you guys are familiar with it, but you have a guide in homology that you need to have when you want to do a true edit, something more than just a knockout. So in order to carry that out, typical operations might employ liquid handling robots and microtiter plates to get the reagents, the guide and the homology paired together in wells to carry out the reaction and the editing, all right? So that's fine, it works, but it doesn't really scale because how many partitions can you have in the long run when you're working in microtiter plates? Another solution is, at the extreme form of this, is to think of the cell itself as a partition, right? So if you deposit the guide RNA and the homology arm together in the cell, along with, say, a barcode, and you tap the full potential of all of those millions or more of cells, then you can do editing at a much larger scale, all right? And the way this is actually carried out in practice is to synthesize these sequences so that they are covalently attached in the manufacturing process. So now every time you deliver one of these molecules, the proper guide and homology pairs are together. If you didn't do this, the chances that the right guide and homology would come together randomly are very low at high plexity, okay? And then the other thing is being able to synthesize these molecules at scale is possible with microarray technologies. So the approach is actually quite elegant in that you can manufacture lots of these sequences and then go through a high throughput cloning and transformation step, and then carry out the editing process. And something I want to draw your attention to here is that we actually use our own proprietary enzyme that we call MAD7 enzyme for the nuclease. That has very good performance in our hands as well as others, and importantly is given to you and actually given to the community free access to own your inventions and discoveries, right? So there's no additional terms to the agreement of using that enzyme. You're free to take the products of your inventions, okay? So this strategy and demonstration of it was first mapped out by one of our co-founders, Andrew Garst, and published in 2016 in Nature Biotech, and has been the subject of some very nice follow-on work from leading labs that are out there that were inspired by it to run with it further. Okay, so once that tube of edited cells comes off of the editing process, you then, like anything else, have a choice to decide how you want to phenotype it. You can either drop into a pooled setting where you're looking at, say, cells, edited cells that are competing with each other, or you can go into, say, a screening campaign. Where you are indeed isolating the individual variants and then characterizing them with usually some sort of typical high throughput analytical method. But at the end of the day, you want to sequence and then combine the genotype and phenotype information to mine it for all it's worth to take it into the next round of evolution that we sort of laid out earlier before. Okay, so enough kind of theory and other examples. Let's actually start sharing some real data. And in this case, we're starting with some demonstrated edit types. So here we're showing some real edits that were pulled out of some of these edited cell libraries. To be clear, these were not done in an isolated way. These were done in a setting where all of the edits were able to be carried out in a pooled format. We isolated individuals from the libraries, and we actually characterized them genotypically to ensure that they had the edits that were in the population. So here we're looking at a promoter RBS insert that's actually a 61 base pair insert with sort of clean reads all the way down, other than a few maybe sequencing errors. And then another example is a 9 base pair swap that confers a triple stop in order to get a loss of function in the genome. And then the final example is sort of a typical saturation mutagenesis library where you're targeting one position in a protein and you're looking to confer all of the amino acid changes that are possible at that position. And then, of course, it's an easy extension to just run something like this across the whole gene. Okay? So that's hopefully a good idea of what the edit types are that are possible. And we'll actually use all of these edit types in the examples that I'm about to show. Okay, so the example is from lysine biosynthesis. Probably many of you have seen this over the years. Lysine, I think, is expected to be like a $7 billion molecule. It's used as a feed additive by 2020. And so we are not a lysine company, but we want to get this out there to try to stimulate you to and inspire you to think about applications in your area that you're interested in. So we undertook a very aggressive strategy to generate diversity in the lysine pathway. And in addition to that, went genome-wide because that's where we really want to allow folks to leverage the technology. And so in addition to hitting the 19 enzymes that we thought were implicated in lysine production in E. coli, we also went genome-wide with 2 different versions of knockouts at different positions in every protein in E. coli, as well as putting a promoter ladder in front of every gene in the E. coli genome, all right? So the total number of edits we were able to design and create were 200,000. So this is really unprecedented, right? This kind of capability is heretofore unthinkable, right? This is really transformational editing capability. This allows you to now go after the full genome with non-trivial edits to optimize the phenotypes or do discovery depending on your interests, all right? So let's walk through a couple of different ways that we phenotyped these libraries. One was through a sort of classical screening exercise, and here we're focusing mostly on the diversity generation part of the exercise, all right? So what I'm showing you here is a box plot of the different libraries that were created and then put together into this big superset of 200,000 edits. And you can kind of get a sense for the size of the knockout libraries, the many different ways we interrogated the pathway with amino acid changes, as well as this promoter ladder that had 5 different promoters of varying strength from the iGEM database, okay? And we screened these libraries, and here on the y-axis you're looking at log2 fold improvement that came out of the primary screening. So we have some things that look like they're dozens or hundreds of fold improved over the wild type E. coli genome. So we started with the very base MG1655 strain. It was not engineered in any way. So there were trace amounts of lysine, but we were able to get across lots of targets in the genome a significant improvement in lysine. And something that's really interesting about where we're going with this technology and the related ecosystem is that we can now generate in this design, create, test, learn paradigm that we all know and love, where create has heretofore been kind of limiting compared to test. We are now fundamentally going to change that impedance mismatch. So create will unleash a torrent of edited cells that you will have to catch, right, in terms of the downstream phenotyping that you want to do. And so we've invested in some high throughput screening capabilities, and here's one example where we used a rapid fire mass spec where every 7 seconds we could get a bead on the lysine productivity, all right? So numerous hits found in the primary screening. So let's start to look at where those hits are. So first let me start with DAP-A. So anyone who's ever worked on lysine is very familiar with this enzyme because it's a key control point in the lysine biosynthesis, and it's well known to be allosterically inhibited by lysine itself. There have been a number of residues and mutations that have been characterized to break that inhibition, and we actually were easily able to pull some of those out in our screening. So what you're seeing here on the x-axis is the fold improvement in one condition, which is kind of our production condition to identify hits, and then the y-axis is another condition. So you don't always get the same result depending on the conditions. But nevertheless, we found a number of things in the amino acid space that were conferring significant fold improvement. And interestingly enough, out of the genome-wide library, this was not targeted or directed in any way, out of the genome-wide promoter library, we pulled out the strongest promoter as driving high lysine biosynthesis. That promoter was in front of DAP-A. So it stands to reason. It's probably not that shocking, but it was a nice demonstration of our ability to find things that were obvious. What about all the things that aren't obvious? So I've been stressing the importance of being able to go genome-wide because we don't know what the rules are for biology. And so what we're seeing here is, and it's purposely sort of meant to overwhelm you a little bit in terms of like the variety of genes that are identified as well as the edit types that were important for lysine production. So you can kind of run your eye down the list of all the genes that are showing up as relevant, as well as the different edit types, frame shifts, promoters, stops, and so forth. And what's interesting is that some of these make sense, and we did find a number of genes in the pathway, in the lysine pathway, that were important for lysine biosynthesis. But we also found a number of targets that have never been characterized before. In fact, some of them don't even have annotations. We all know the Y-ome in E. coli is largely unannotated and uncharacterized, and we found a number of those. Other targets are annotated, but they're things like secondary metabolite degradation. They don't, they wouldn't show up high on a list of rational targets, and yet we were able to pull them out, okay? So that's the screening campaign. But as I kind of mentioned earlier, we don't have the capacity to look at everything in the screen. We actually only dipped our toe in the water. We only sampled about 18,000 or so. We plan to go back and do more deep screening based on the juicier libraries. But we wanted to apply a selection-based approach to see more examples of edits conferring a phenotype of interest. So here what we did is we set up a sort of a standard selection. In this case, it's an anti-metabolite selection process where we challenge the cultivation with an anti-metabolite called AEC. And it looks like lysine, but for a sulfur that really mucks up the metabolism of E. coli. So what you're asking the bug to do is to produce more lysine to outcompete this toxic inhibitor. So we went ahead and introduced AEC into some of our biological samples. So here what you're seeing is control and treatment, so 0 or 3 millimolar AEC across 3 biological replicates. And you can kind of even see visually some partial clearing in the cultivations that had AEC. So there's definitely an impact when you apply this AEC. The question is, what's in those flasks, right? What's getting enriched? So let's take a look at that. So what we're looking at here is a scatter plot for the two different pairwise samples in every way you can compare controls and treatments. And what you're seeing is for the control samples, and these are counts, log 10, so you're getting up into the thousands or tens of thousands of counts for each condition. And what you're seeing is for the controls, you're seeing very high biological reproducibility. And each of these data points is individual editing cassettes. We have an Inscripta barcode editing process that gets subjected to NGS sequencing, and we can track the population of those editing cassettes. And so you see a count in one sample looks like the count in the other sample for the same condition. Same result is true for the treatment condition. You're seeing lots of good biological reproducibility. So this gives us confidence that if we see a difference between treatment and control, it's likely to be real. And in fact, we do see some significant differences. So in particular, we're interested in regions that are enriched in counts in the treatment condition and are modest in counts in the control condition, right? So let's double click on these kind of enriched populations. And again, use DAP-A as a starting point, not to get overwhelmed. And what you see is a number of amino acid changes that are reasonably high counts in the control condition, but are depleted in the treatment condition, right? So these are things that are likely affecting lysine production, right? And in fact, one of the things that you see are deleterious is putting a low-strength promoter in front of DAP-A, so probably not surprising. On the flip side, we see a lot of the same things we saw in the screening campaign, where we see lots of mutations in DAP-A that are enriched in the population, 4,000, 10,000 or more compared to dozens or hundreds in the control, okay? In addition, we find a number of promoters in front of DAP-A that are conferring selective advantage, okay? So that's great, but now let's step back and look genome-wide. And what we see is some really interesting results. So if we start with the most enriched edit within the subset of just the pathway amino acid changes, we find that each of the enzymes in the pathway had some ability to confer some survival benefit in the treatment condition. 3A and DAP-A, again, were sort of the big winners. But now if we look genome-wide, we have genome-wide knockouts now. Again, completely irrational, just let nature tell you what works and what doesn't. And we recapitulated actually a known target that was found in an earlier study called Lysp, which is a lysine transporter, but it's also involved in regulation. It's a little bit of a mystery how it works because it's a knockout. And then if we look at the promoters, we have even more beneficial diversity we found. So another 91 targets that were found through at least one of the rungs on the promoter ladder, all right? So in total now, we have 100 new targets identified outside of the lysine pathway that we can use in a combinatorial search. So briefly, I sort of had mentioned with DAP-A, the correspondence between the screen and the selection. And so we looked at all of the instances where the screen and the selection had the exact same edit in the population. And what we find is fairly good correspondence, especially given their very different systems. So you're finding things that are highly ranked in the screen were also highly ranked in the selection, and vice versa. Okay. So let's just pull on a few little threads or vignettes here. So one of the things you want to know is, are you driving biology in ways that you predict? And so you can look at knockouts and promoters as sort of two sides of the same coin. So what we're showing here is for a few hand-picked examples, if you use a knockout strategy, you get enrichment, whereas you put strong promoters on those same genes, you get depletion. And here it's a reversal of fortunes where the knockouts actually reduced the presence in the treated population, whereas these promoters were able to confer a strong enrichment, all right? So that's just a few, and you could cherry pick, to conform with your preconceived notions. But if you want to look genome-wide, that's thousands of dimensions. How do you characterize that? Well, a typical approach to do that sort of thing is principal component analysis. And what I'm showing you here is in this three-dimensional PCA subspace projection, you're looking at how the two knockouts compared to each other. So this is across 3,600 genes that had enough counts. They cluster together, which is not surprising at all. And then you see some interesting behavior among the promoters. The highest strength promoter is out here. It's a little bit on its own. The very weakest promoter is down here, and then the middle promoters are clustered here. But it's worth stressing that the first principal component completely separated the knockouts from the promoters. So it's a nice sort of just easy verification or validation that you're getting meaningful, you're driving biology in meaningful and predictable ways. But one could imagine mining these kinds of data for new insights. Okay, so I talked a lot about, the ability to recruit beneficial diversity. So let me just show you the beginnings of what the story looks like when you start stacking mutations. So we had identified, and others had identified, an important position in DAP-A, again, that was important for likely breaking allosteric feedback regulation. And that one change was several hundredfold improved over the wild type. And then that strongest promoter that we pulled out was close to 1,000-fold. So if you build a library now on top of this slightly engineered strain, that high-strength promoter library confers additional benefits. In fact, we found things that were even 1,500-fold improved. And if you just combine these two edits, you get what is typical, what we've seen a lot of times in protein engineering, is an almost multiplicative effect. So we now found something with just two edits that's 10,000-fold improved over the wild type. Okay, so kind of like pulling it all together, what is it we're trying to do in Inscripta? What's the vision we're aspiring to? And I just want to go back to the early cartoon that we had about what we're enabling. And the idea is we want to break away from this serial mutagenesis process. We want to be able to offer tools and strategies to really develop highly parallel, massively parallel single editing capabilities to generate diversity, and then based on that diversity, be able to recombine it in ways that allow you to move faster through the phenotype space. All right? So just an early look at what that is going to be for E. coli and yeast is some data showing our ability to confer these kind of large-scale edits across the genome now in multiplex combinatorial formats, right, where you're loading up the E. coli or yeast genome with multiple edits, 2, 3, sometimes 4. Okay? So in summary, I hopefully gave you some understanding of how we can use all of the great lessons and strategies that have been developed in proteins over the years, namely diversity generation and combinatorial search or optimization. And if you combine that now with the tools that are becoming available that include unprecedented scale, superior performance, and greater access, then we believe that's going to allow you to create and test a lot more ideas, discover and exploit new biology, and probably most importantly is allow you in the research community to be able to focus your expertise and resources on your own questions and goals rather than struggling with the technology to confer those genomic changes. All right? So, Inscripta. We are developing the world's first scalable platform for benchtop digital genome editing. So we're currently in early access with some global key opinion leaders. The technology will be commercialized this year for E. coli and yeast, and we intend to offer both single and combinatorial editing formats. And the platform will consist of software, instruments, and reagents, and the instrument in particular will be push-button and fully automated. So we want to hear from you. We have some ideas of how the technology could be used, but we are under no illusions that we have the market cornered on all the things that this technology is going to open up. So we are just at the beginning of starting to engage the community, and we're very happy and humbled to be able to share with you today at SEED, really for the first time publicly. And so if you're up for joining us for the poster session, and if you have any questions, we have a poster describing some of the things that you saw here today. We'd love to hear your feedback, thoughts, and ideas. And thank you for listening.